The overall goal of this procedure is to observe the partitioning of viral genomes in dividing cells. This is accomplished by first making a plasmid that can be visualized by fluorescence microscopy. Here, a plasmid is engineered to include tandem repeats of the lactose operator sequence, or LAC-O. The second step is to introduce the plasmid into dividing cells, followed by infection of the cells with a retrovirus encoding the lactose repressor, or LAC-I, fused to a fluorescent protein. The lactose repressor fusion proteins will specifically bind the lactose operator sequences in the plasmid. Subsequently, the cells are monitored by fluorescence microscopy to track individual plasmids through multiple cell cycles. Ultimately, the images acquired are processed to determine how the viral genomes are maintained in dividing cells.
The main advantage of this technique over existing methods such as quantitative PCR, southern blotting, or fluorescence in situ hybridization is that this technique allows individual plasmids to be observed at multiple points in time.
To enable visualization of the plasmid by fluorescence microscopy, standard molecular cloning techniques are used to introduce a DNA fragment containing approximately 250 copies of the lactose operator sequence, or LAC-O, into the plasmid. In this example, the plasmid is a backmid of approximately